Okay, I want to welcome you to the panel on origin and evolution of sociality. I'll keep my introduction really short because we have a panel of really excellent people, and they, I'm sure, make a better job explaining the advances we made in that field and the questions which are still open in the field. A little teaser here: sociality is quite widespread, not just in humans kind of have the tendency to really just have others as a center, but humans are not really the pinnacle of social evolution. The pinnacle in mammals, maybe on the bottom side, the naked mole rat. There's microorganisms which show quite complex social behavior, which we'll hear about today, and of course a lot of the social insects, which we kind of study extensively here at ASU. Brief intro into the panel. So we kind of lost one of the speakers. He spoke in the panel before. Nessie was supposed to speak here. Now we have 4 people. Richard Dawkins will kind of answer or try to answer the questions which Nessie kind of started out, things which are good to have evolved, like altruism. So how can we explain the evolution of altruistic behavior in the face of the selfish gene, or 20,000 selfish genes? Joan Strassman then will talk about the origin of eusociality. She has done a lot of work on polistis, more what we social insect people would say more primitive social systems. And she has also done lately a lot of work in slime molds, and they are social. Yeah. Bert Feldobler then will talk about cooperation and conflicts, 2 intertwined themes in social evolution. He just published a book on superorganisms, or the superorganism concept, and that will be probably an interesting point to make. And last not but least, Dave Queller will talk about the major transition in social evolution. And without any further ado, we'll go to our first speaker, Richard Dawkins, who will talk about how can we explain the evolution of altruistic behavior.
The title I was given was Darwin's Dilemma, how can we explain the evolution of altruistic behavior? I suppose I'm the one who has to set the background, selfish genes 101 or something like that, before we get on to the new stuff. So I apologize for that. I'll try and rush through it. Darwin considered the evolution of altruism in humans, and that was the only place where he resorted to a form of group selection, sort of intertribal selection. I don't want to talk about that because most of my colleagues here are talking about either social insects or similar problems, so I'm not going to talk about humans. In the social insects, Darwin didn't specifically talk about altruism. What he talked about was something very similar, which is how sterile worker castes can evolve their capabilities. Since they're sterile, they can't actually pass on to the next generation the qualities that they have to make them effective workers or soldiers or trapdoor spiders, ants or whatever they are. So Darwin faced this dilemma, and he got the right answer, not surprisingly. He said, he used an analogy from domestication. He said, thus a well-flavored vegetable is cooked and the individual is destroyed, but the horticulturalist sows seeds of the same stock and confidently expects to get nearly the same variety. Breeders of cattle wish the flesh and the fat to be well marbled together. The animal has been slaughtered, but the breeder goes with confidence to the same family. I have such faith in the power of selection that I do not doubt that a breed of cattle, always yielding oxen with extraordinarily long horns, could be slowly formed by carefully watching which individual bulls and cows, when matched, produced oxen with the longest horns, and yet no one ox could ever have propagated its kind. Well, that in a nutshell is the answer to the riddle of social insects. Nowadays, we'd put it in language that Darwin wouldn't have used, because Darwin didn't know the language of genes. We would put it in the language of genes, and we would indeed put the whole of natural selection in the language of genes. Since the neo-Darwinian marriage of Mendelism with Darwinism in the 1930s and 40s, we've become accustomed to thinking of natural selection in terms of changes in frequencies of entities that you can count, changes in frequencies of alleles. So if we're going to talk about adaptation, if you ask the question, what entity in the hierarchy of life is the adaptation good for? The answer is the gene, because the gene is the entity that passes from generation to generation, potentially immortally. The world becomes full of those alleles that are good at staying in the world, and that means good at making bodies, or making phenotypes more precisely, which preserve and propagate them. And the reason why that works is that on the whole, genes ride inside the phenotypes that they have created, and therefore their fate is bound up with the phenotype, usually the individual body that they've created. Now, because of that, because that's the way it works, people got extremely accustomed to the idea of animals working for the good of themselves, individual animals working for the good of their own reproduction, themselves and their offspring. And so a language grew up of fitness. The fitness of an individual organism was something like its capacity to survive and produce not just offspring, but grand offspring, great-grand offspring, a very large number of descendants, an indefinite number of generations into the future. So people became accustomed to discussing this at the level of the individual organism, and individual organisms maximized something called fitness. Then in the mid to late 20th century, it began to be realized that, of course, offspring are not the only individuals who carry the genes of the parent. So in the same way as we can say that a parent shows altruistic, caring behavior towards its offspring, because the offspring have a statistical probability of sharing the same genes, the same applies to other relatives as well. And you can easily calculate it statistically what those odds are. The odds of a full sibling sharing the same genes are 50%. That has to be qualified. It's not literally the same genes. It's the same. There are two ways of looking at this. Either the same genes that are rare in the population as a whole. One has to say that because of the fact that all of us share more than 90% of our genes anyway. So we're talking about the sort of cream on the top, those genes over and above the ones that are shared anyway. That's a very crude way of putting it. But a simple way of putting it, which kind of works but is not adequate, is to say it works for rare genes. A new rare gene in the population will tend to be shared by 50% of close relatives such as siblings or offspring, 25% of half-siblings, nephews and nieces, etc. So Hamilton formulated the rule that, so-called Hamilton's rule, that a gene for altruism will spread if the benefit to the beneficiary is greater than the cost when the benefit is devalued by a fraction, it's called the coefficient of relationship, which is a half for full siblings and offspring, a quarter for uncles and nephews, etc. So a gene will spread if RB is greater than C. Hamilton chose to express it at the level of the individual organism, and he coined a term, inclusive fitness, which was a modification of the traditional fitness. And I told you what the traditional fitness was. It's survival and production of a large number of descendants in the indefinite future. Inclusive fitness is a more complicated function, and Hamilton calculated how to do it, and he got it right, and it was a great breakthrough in theoretical biology. Inclusive fitness is informally defined, and Hamilton confirmed that he didn't mind this informal definition. Inclusive fitness is that quantity which an individual will appear to be maximizing when what is really being maximized is gene survival. So the easy way to get your sums right in this field is to pretend that you're a gene. Jacques Monod did this. Jacques Monod once told me that when he's trying to solve a chemical problem, he said to himself, what would I do if I were an electron at this point? Well, the way to get your sums right in discussing the evolution of altruism, or indeed anything else, is to say, if I were a gene, what would I do in order to get myself into future generations? And one of the ways you get yourself into future generations is to cause your body to look after close relatives or less so, more distant relatives, and so on. That's an extremely inadequate summary, but I've run out of time.
Thanks, Richard. We'll go to the next talk. From John Strassman, and she will talk about the origins of sociality from a WASP standpoint of view.
Maybe one of the most interesting origins for sociality is something that we see in the social insects, and I'm going to focus on particularly on eusociality. I realize this is quite a diverse crowd, so forgive me if this seems too simple. First of all, what is eusociality? This is not the textbook definition, it's the working definition. It's simply species that live in groups in which many individuals do not reproduce and instead help others to have more young. So we are not a eusocial species. By this argument, you might argue, if you want to go higher or lower, that we are a not very highly social species. Well, what is? Some wasps, not all wasps, there are also solitary wasps. Some bees, here I show a honeybee and a stingless bee, there are also solitary bees, so not all bees. All ants, so ants are not good for looking at the origins of eusociality because they're all eusocial, as are all termites. Naked mole rats is this, we all seem to have the same picture even. I think it was actually off a porn website, so I don't know what that says about us. And then there are some other newer social organisms like aphids and beetles and shrimp and things. So why is eusociality so hard to explain? Well, that was really treated already in the first talk. I'll just summarize here. Behaviors evolve because the genes that underlie them become more common over time. A behavior that results in helping others is therefore hard to explain, although, of course, Richard already explained it. Okay, so how does eusociality arise? Again, this is Hamilton's rule, which was already introduced to you. I'm going to use it here in a little bit of detail. It's called kin selection often, and I'm becoming more and more appreciative of the fact that this isn't a great term for it, because it focuses attention on the relatedness factor. So people will often, instead of doing the careful inclusive fitness calculation, simply say that individuals should help others in a linear relationship to their relatedness to them, which is incorrect. In fact, you should help if you pass on more genes by doing so. And so to look at that, you have to know what you're relinquishing in terms of individual progeny or fitness that you have by rearing your own young versus helping someone else. And you don't get to count all the babies that someone else has, only the ones that they have only because of your assistance. Now, there was a lot of excitement early on in the social insects because they're haplodiploid. Males are haploid and females are diploid. So dad gives the same sperm, genetically speaking, to every single one of his daughters, which means that they are related to each other by 3/4. So there was a lot of excitement that this might explain the commonness of helping and eusociality in the ants, bees, and wasps. In fact, it does not. If you simply go out and measure relatedness, and this is for a bunch of these primitively eusocial wasps that do not have morphological casts, individuals are totipotent, but in fact, many of them simply help others. You can see that relatedness is, in most of them, well below the 0.75 relatedness level, and these are 95% confidence intervals, not standard errors. Well, there's other reasons why eusociality may be so common in the Hymenoptera. First of all, parental care is ancestral to the social wasps. The mother wasp will grab a caterpillar or a spider or something like that in many solitary species, sting it, lay an egg on it. So she's got both a sting, which can be used for prey or easily used for defense, and a certain level of maternal care. So these other factors can explain the commonness of eusociality in Hymenoptera. This failure of the haplodiploid hypothesis of exaggerated relatedness doesn't mean that kin selection itself is not important. But it kind of changes the question. So what we then want to know is, well, how can you produce more siblings than offspring? What's going on? What is the fitness environment that causes this sort of thing to happen? And I'd like to argue simply that there are two general cases where this happened. First, if you nest in a highly defended place where you can also eat, where it can be expanded, if you stay there and help mom, rather than take the risky step of going off and being vulnerable to predation, you may simply be able to rear more young. And this is an explanation we think is general for things that live inside their food: termites, galling aphids, thrips, the naked mole rats, social shrimp that live in sponges. We think this is quite common for these. And the first caste to evolve in this case is soldiers, which often develop ways of defending: horns, jaws, claws, things like that. And then the second one, which is nearer to my waspy heart, is the life insurers. And here the idea is simply, if two stay together, if you stay with mom to rear babies, then if mom or you die during risky foraging endeavors, the other one can finish rearing the young. So your fitness can take the half-started young. And in these cases, we think foragers will be the first form to evolve. So these are two sort of general cases. I'd like to argue that just using kin selection as a framework in an ecological environment, not just looking at relatedness, we can see what's going on. Individuals must be related to those they help, and they can pass on more genes under certain circumstances by helping than by striking out on their own. I haven't talked at all about my current love, which is the social amoebae, and yet I think that similar arguments, though different ecological ones, could be made for them. Well, this is very far afield from what most of you do, so you might think, well, why should we care about this kind of stuff? Bert will talk about how looking at social insects and the evolutionary conflicts manifested in behavior allows us to come to theories that have proven general. And then we'll end up with Dave Queller, who will show how this generality of conflict has been important in the elaboration of life. Thank you.
Thank you, Joan. Just go to the next speaker, which is Bert Haldobler. And John has already introduced him.
Can we dim the light a little bit for the slides? Where's this gentleman?
Is this speaker?
Well, anyway. Well, the evolution of eusociality is indeed one of the great transitions in evolutionary history. And only a handful of species, in a way, groups did this, but they play an enormously powerful role, dominant role in most land habitats, most land ecosystems. Now, the ants, as we just heard, are all eusocial. There are about 14,000 species of ants known. They make up only 2% of all insect species, but they comprise 70% of the entire insect biomass, and about 20 to 30% of the entire animal biomass. So why is that? only 2%, and such an enormous biomass? Well, it has something to do with eusociality, because eusociality means or entails a complex division of labor system. Now, we study the ants particularly, not only because they are socially or ecologically so important, because they give us not the hint, as Jones showed us, how eusociality originated, but they show us the diversity of eusocial systems. And we can look at different evolutionary grades of eusocial systems. And I give you two examples, two case histories. One is a very primitive eusocial system, and one is a highly advanced, what I like to call a superorganismic eusocial system. And as you will see, the cooperation and conflict plays a major role in the evolution of these eusocial systems. Now, the first, can we dim the lights a little bit? There's a gentleman who wanted to dim it. Here you see actually a primitive Pomerine ant. This is the queen caste, these are the workers. And what you immediately recognize, there is not a large morphological skew. The workers have almost the size, by the way, the dots we did. They have almost the size of the queen, and when you dissect the workers, you see these workers have well-developed ovaries, and many of them are mated. 80% sometimes in this colony, the workers are mated. Why is that? The queen in these primitive ants have a relatively short lifespan. They live only for 2 to 3 years, and then the whole colony would actually also die, but not in this species. These workers have all the full reproductive potential. So we call them workers, but in fact they can be full reproductives. They cooperate still as workers. However, when the queen is gone, you see enormous conflict breaking out in this society, and competition and conflict within the society is prevalent. You see ritualized combats which can exaggerate to real wrestling fights, and actually in different forms and shapes. And finally, some winners crystallize out, and these are the ones which lay the eggs. Now, this group is usually a group of so-called workers. We call them then gametokrats, because these are mated egg-laying workers, gametokrats, married worker. So there is usually a group of those which made it up to the aristocracy of reproductives, but they are continuously monitored and checked and challenged by younger workers. And there is clear monitoring going on, and Jürgen Liebig, actually, who is here in the audience, this is work done here at ASU, was wondering, how do these ants realize that some of the workers begin to wane in their fertility, and though they see an opening to make it up to the aristocracy? Well, it turned out that these workers indicate their fertility state by a complex hydrocarbon patterns on the cuticle. These are honest signals, and I cannot go into the details, but what you see here, I just show you here discriminant functional analysis, and you see here, these are all reproductives. The blends of these hydrocarbons, they are statistically significant separated from here, from the young workers. They still may make it. They may actually replace some of those and then enter this top group. And the old workers, which are basically outside workers now, and which will never make it again to this reproductive group. So as you see, the individuals in such a primitive eusocial system are expected to resist evolutionary specialization to become workers. And naturally, in these systems, the division of labor among workers is relatively poorly developed. German word slipped in. And the communication system is relatively primitive. Now, when we look at these societies, and each one here, each circle is one of these colonies, and each dot represents an individual. You see a lot of friction going on within the colony competition and aggression, but actually very little competition and aggression between the societies. They have no territories, there's almost no interaction, they're individual foragers, they avoid each other, but no between colony competition. So there is apparently, of course, kin selection has driven this eusocial system, but there is still a great deal of individual level selection going on within the societies. Now let's make a big step to a truly superorganismic eusocial system. These are, and I have chosen the leafcutter ants. They have tremendous nests. If you want to understand how these nests look like inside, you pour in 10,000 liters of water mixed with 10 tons, or 6 tons, actually to be correct, of cement, and then you wait for 3 weeks, and then you excavate the nest, and this is what you find. These are incredible edifices, about 50 square meters in area, going down 8 meters, large subterranean tunnels, the fungus chambers, these are fungus growing ants. So a remarkable structure, which, and I take here a metaphor which Richard Dawkins coined, it is indeed an extended phenotype here of the queen, yes, and her mates. But really, you see here the colony traits. Now, there is only one reproductive, the gigantic queen. This queen now can live 10 to 20 years. All these others are, all these others here you see, they're also females. The morphological skew is tremendous. Now, this queen mates once in her lifetime with about 5 to 6 males and stores 300 million sperms in her spermatheca. It's a sperm bank, in fact. As a male, you die after mating, but you can become father 20 years after you died because they are able to preserve the sperm in tropical heat. A remarkable fact. We don't understand yet how. And they produce, this queen produces in her lifetime about 150 to 250 offspring, a million offspring. 150 to 250 million offspring. This queen, which has such a long lifetime. Her workers, of course, they carry all the genes of her and her mates, but different genes are expressed in this worker caste, subcaste, as you see. There are the minors. They are genetically relatively identical, but different genes are expressed, which make these, of course, different genes than the queen expresses. And these different sizes, these subcastes, are beautifully tailored to do the job, to grow the fungus from which they live. The big ones cut and carry the leaves, and then you have this assembly line going down to growing this fantastic fungus from which these ants live. So when you look above ground, you see they have huge territories with huge trunk trails. This is all a map from nature. And you will see that these trunk trails are defended, these territories are defended against the invasion of neighboring colonies of the same species. So you have here tremendous inter-colony competition and aggression, but when you look inside, there is actually no competition, no aggression going on. It is a so-called really the perfect superorganism. And I would say now that in this case, and it doesn't question the kin selection theory, in fact, actually it supports it, but there is another level of selection going on, the selection on the colony level in these competitive interactions. And again, I borrow a term which Richard Dawkins coined, the colony as such becomes a vehicle of genes. Thank you.
Okay, thank you. We're in a little bit over time here. but we'll give Dave the same chance, not for the overtime, but at least 8 minutes. And Dave Queller will talk about the breadth and depth of social origins.
Okay, why are we interested in social insects in the first place? I started out being interested in them simply because they were an intriguing puzzle that went all the way back to Darwin, and I think you've gotten an appreciation now that that puzzle is largely solved, but we're still interested for a variety of reasons. And one is that there's both a breadth and a depth to the importance of social cooperation in life, and that's what I want to talk about a little bit. First of all, cooperation is more widespread than you might think. It's not just social insects. There are many other organisms that do that. Here's a selection of cooperatively breeding vertebrates. You already saw the pornographic picture of naked mole rats. They're the most famous example, but there are many others as well. Meerkats, African wild dogs. They're much like social insect colonies in the sense that there are a couple of breeders, usually a male and a female, and the others are assisting them in rearing offspring. There's also a lot of conflict going on typically, so they're not as superorganismal as a honeybee, for example. Within invertebrates, there are more than a handful of really nice examples. Many of the familiar invertebrates, such as corals, are actually colonies of individual organisms. Each little blob on that coral is a polyp, a complete animal body. They're interconnected and interact, and they are clonal. They're genetically identical, so it fits very well with Hamilton's rule in inclusive fitness. More advanced than that even is the Portuguese man-of-war, which is also consists of polyps, but individual polyps, but ones which have specialized to an extreme degree, much like, or even more extreme than the worker casts and queen casts of social insects. You've got the floater polyp and the feeding tentacles and so forth, all different kinds of polyps. There's also a lot of sociality, and this was surprising, in the microbial world, and it'd be fun to talk about that at length. As Joan said, both of us are now working on this first organism here, the social amoeba, which forms these fruiting structures where individuals come together, 20,000 of them perhaps, to form a fruiting structure in which 20% of the individuals die, giving up their lives in order to support those other individuals as spores and help their dispersal to a better feeding site. Even bacteria have cooperation. The myxobacteria in the middle there have a behavior a bit like the social amoebae, but also in their feeding stage, they cooperate and hunt more or less. They've been described as wolf packs. They move together in coordinated movements, secrete external enzymes to digest prey much larger than themselves. And even the humble intestinal bacterium E. coli that you're all familiar with is social, at least in the sense that many clones evolve the capacity to kill competing clones and promote the production of their own clone, and do so suicidally, much like a worker honeybee giving up her life. And finally, a huge area of cooperation that we haven't really touched on is cooperation between species. So for example, you've got cleaner fish host symbioses, where a small fish will pick parasites off the skin of a larger fish. And you've got legumes and their rhizobial bacteria, where the plant provides carbon for the bacteria provide nitrogen for the legume. This is a completely different kind of cooperation in that it doesn't depend on relatedness. Obviously it can't. These are separate species. They don't share the same genes, identical by descent. Instead, it depends upon mutual benefit. They provide something for each other, but you still have the same kinds of conundrums that we had for kin alliances too. You have the possibility of cheating. What's to keep a cleaner fish from occasionally taking a bite out of the fin of its host, or the other way around, what's to keep the host from occasionally making a meal out of the cleaner fish? You have to have evolved mechanisms which solve those issues. So that's for the sort of snapshot of the breadth of evolution, but the depth is in some sense even more important. We've seen how social insects can evolve what Bert called superorganismal colonies. I would in fact call them organismal. If you've got a honeybee colony, the colony really functions as an organism. And part of the reason I would say that, and not use the word superorganismal, is because we've had these kinds of transitions before. You are each an organism that is in fact a superorganism, that is built out of components which were initially themselves, ancestrally themselves, separate organisms. Separate cells, for example. So we've all evolved from a single-celled ancestor, the mechanisms are complex and interesting, but in terms of the evolutionary problems involved, in terms of social evolutionary problems involved, it was not particularly complicated because all of us are multicellular clones derived from a zygote. And so relatedness is extremely high within our own bodies. The cells are genetically identical. Of course, they can evolve to help each other. In nature, there are always exceptions, and that's one reason why we're studying this little social amoeba I'm showing you again here, Dictyostelium. Those form their multicellular structures from separate cells coming together, and so you have the potential for genetic chimerism where you can have conflicts within that structure. In fact, they are often clonal, but sometimes not, and you get interesting conflicts. Going down a level further still, starting from a single cell, the cells that make us up, in fact, are superorganisms themselves, or organisms at a higher level. Way back in the history of the cell, a host cell engulfed one or more bacterial cells that became organelles in our eukaryotic cells. So mitochondria, the powerhouse of the cell, are derived from bacteria that were ancestrally engulfed and became a part of that organism. In plants, chloroplasts also had a similar origin. So you have, this is one of these mutualistic major transitions that was extremely important in the history of life. And finally, if we go all the way back, well, not all the way back maybe to the beginning of life, but very close to it, we've had some talk about the chemistry that has to be involved and so forth, and I'm no expert in that. I don't know about the primordial soup very much, but I want to talk about the primordial group. Even at the dawn of life, when replicators were almost naked replicators and not very complicated, you had these social conundrums. How do you cooperate for the greater good? So in this diagram here, each of these little bars represents a replicator, maybe a bit of RNA in the RNA world, which can both replicate and perform some little job, maybe a job essential in the step of replication. And initially, if they're not enclosed in a membrane or something, the jobs that they perform are sort of available to everybody. And if you had one particular replicator, the red one there, which I've labeled as a selfish non-producer, all it does is serve as a template for replication. It doesn't spend any of its time doing a job. It's going to out-reproduce the other ones. At the end of the arrow, you get more of the red guys. You get more non-producers, fewer producers, and this can be disastrous for cooperation. One solution to that is enclosure. You see that in the bottom. If you enclose replicators within a membrane so that they interact only with themselves, the ones that happen not to have the selfish red one out-reproduce the ones that have the selfish non-producer. Another important transition there is the putting together the replicators into a chromosome. So you still have the problem in the cell that perhaps the one will be a selfish over-replicator that makes more copies of itself. So in the daughter cells, you've got more of the blue replicator than the others. Ultimately, this could lead to having all the blue ones, and some of the jobs of the cell won't get done. If the replicators instead are strung together in a chromosome and replicated all together uniformly, then you reproduce them heritably and don't have over-reproduction of those types. So I just want to conclude by leaving you with a couple of thoughts. One is, well, Antonio Lascano said that life is living things are a lot like politicians, and I think that's true, more than the sense that they're just optimistic. Living things are sort of slimy bastards that are self-seekers, and yet they do their self-seeking often through cooperation. And it's really interesting and really important to understand the roots of that cooperation and how it's led to some of the major transitions that we see in life. Thanks.
Thank you. Thank you for all the contributions. We have very little time. I think what came through from all the speakers is that really kin selection or inclusive fitness concepts is kind of the underlying dogma. Without we would not understand the evolution of social behavior. But the question is still, why are some organisms social and others not? And is the kind of now levels of selections, or however you want to label this particular concept, like superorganism, taking away from the gene perspective, will that help us to explain why some organisms are social or not? Richard, will you want to give a stack of that?
It's always an interesting question to ask why some animals evolve something and others don't, because the same pressures in some sense are pressing on all of them. If you take Hamilton's rule, a gene will spread for altruism if RB is greater than C. We mustn't neglect, as Joan said, all three of those terms. We mustn't concentrate on R to the exclusion of the others. R is important, and despite what she said, I suspect that haplodiploidy might have had some important influence. However, B and C are also important. And the other things that she mentioned, like the advantage of staying behind in the parental nest, perhaps because it's hard to find a new nesting site, that is going to bias a particular species in the direction of helping at the nest in some form. There are going to be lots and lots of ecological factors of that kind which will tend to bias a species to go for the helping at the nest solution rather than the going it alone solution.
Dave?
I think I could add just to reiterate some of what Joan said, that it's not just ecological conditions, it's also the sort of pre-adaptations that organisms have. The Hymenoptera just happened to have a suite of traits that were suitable for parental care, and those could be easily co-opted in the context of alloparental care, giving care to somebody else. So it's a combination of ecological circumstances and where the organism is in its evolutionary history at that point, I think. I don't think we're real good at being predictive about this yet, but I think perhaps getting better gradually.
Yeah, when Bert gives a stack, I mean, I have time for 3 or 4 questions, so when the mics can go, if you raise your hand when Bert gives his answer.
I just wanted to say what is often forgotten, that actually parental care is already part of the whole inclusive fitness concept, and this is often misunderstood. And so this is social behavior. What is rare is eusocial behavior. And this is, but I mean, including my good friend, Ed Wilson, misunderstood this for a while, and I have to admit I too, that parental care is really a part of the whole Hamilton's inclusive fitness. And I wanted to say one word. I just before Hamilton went to the Congo, he visited me in Würzburg, and he said to me he never liked the term kin selection because I jokingly said it is because Maynard Smith coined it. You know, they didn't like each other very much, and he said no, it is misleading. He mostly used inclusive fitness, and I think I have gone over now and always call it inclusive fitness theory, because it narrows our perspective, the pure kin selection concept.
Okay, one question here.
Throughout the discussion, I was itching to ask a question that David Queller touched on at the end, which is that the most dramatic example of cooperation that we see in biology is multicellularity. And I just wonder, we sort of leapt over that stage in this great origin story, and now we're on to sociality and related concepts. Does this provide any insights into the origin of multicellularity? How did it emerge, and what led up to it?
I think the simplest insight I alluded to already, maybe I'll repeat for lack of deeper insights perhaps, and that's that most of the multicellularity that we see is clonal multicellularity. The relatedness is extremely high. It's as high as it can be. Things are genetically identical. And I think it's interesting.
Yeah.
I think so, yes.
Okay, one brief last question. I'm ordered to end at 4:10. Yeah.
Yeah, at the other extreme, evolutionarily, why do some organisms eat their young?
Great resource, I would say. So, I mean, killing of the own offspring can be adaptive in situations where the resources are scarce, and so if killing your own offspring increases your probability to kind of have successfully rearing a next litter, then you should probably do that. And we see a lot of those examples. Mice, for example, will actually dissolve their embryos if you stress them too much. Okay, I need to stop here. Thanks again for the great panel members here, and I think we have 10 minutes time.
No, rapid turnaround.
Okay, rapid turnaround.
story. I won't be long.
Good job. Look good.
Yeah?
I think we have 5 on this one, right, Megan?
And indeed, when you look at his
6?
Curtis needs to be here. He's got the computer. You can be wherever you want otherwise.
An additional seat in there.
Ja, collega.
There's 6 people.
In the back row. It's a PC too that works.
Do you have a pointer, Bill?
Yeah, we're just seeing if I can get it on.
Is the pointer about?
Oh, what are you going to point at?
Who's up first?
I am.
Is there a pointer?
Don't if you have nothing.
Otherwise, the
Lawrence.
All right, you're right. That's fine.
I have a pointer.
Okay, good.
Every time you have 5 people,
Yeah, good.
okay, so we're going to go to
5:35. Okay, cool. Okay, welcome everybody to the final session of today, the one dealing with human origins. And so we arrive now at the twig on the tree of life reserved for the species Homo sapiens. And there's no field with more transdisciplinarity than human origins, which links together genetics, paleontology, archaeology, comparative functional morphology than human origins research. And that's reflected on the composition of our panel today. And so rather than ask each of our panelists to address one specific narrow question, we thought it would be more provocative, interactive, and maybe fun to ask them to respond each to the same question, which is, from the perspective of your research area, what does it mean to be human? And we'll start with the evolutionary geneticist and evolutionary ecologist Alan Rogers. And then we'll move to 3 presentations by individuals who address human origins from the point of view of either the fossil record or comparative primate biology, Don Johansen, Ian Tattersall, and then John Flegel. And finally, we'll finish up with Curtis Marion, who conducts archaeological research on modern human origins in Africa. And so we'll go right to Alan.
If I had tried to give this talk 10 years ago to tell you what evolutionary genetics had to say about modern human origins, it would have been all about population size and the history of population size, about how long it had been since various things happened in our history. And today, as you can see by my title up there, or I guess maybe you can't, but anyway, today none of that stuff is going to play any role in what I have to say. And the reason is that we have wonderful new databases now that are available to us that tell us all kinds of wonderful things, but because of the sort of subtle biases that have crept into the selection of individual polymorphic sites as they have been selected for inclusion in these databases, they tell us very little, in my view, about the history of population size. So we, as a field, have made lemons out of lemonade. And since the new databases don't tell us too much about the history of population size, we have studied what they do tell us about. And to give you a sense of how this works, I'm going to start by taking you through a slide that, well, let's skip that in the interest of time.
Skip it?
Yeah.
Okay.
I'm going to take you through a slide that shows you a sort of a view of a single piece of a human chromosome. Happens to be the portion of the human chromosome that provides the code for one of the dopamine receptors in our bodies. But anyway, each row in this thing corresponds to an individual human being, and each column corresponds to a single polymorphic nucleotide site. And the color codes tell us what the individual human being, what genotype that individual human being had at that genotype. So the dark blues are, those individuals had the common, or were homozygotes, had 2 copies of the common allele at that genotype. The yellows are homozygotes for the rare allele, and the reds are heterozygotes. And so what I'll encourage you to notice in this is that there are kind of 2 big blocks in this data. There's a big block down here that consists of a whole lot of humans that had nearly the same genotype at a large number of sites. It's as though someone took a single chromosome and made a whole lot of Xerox copies of it. That's not my metaphor. I don't know who invented that. But anyway, this looks like a whole bunch of Xerox copies of the same chromosome. And this part up here doesn't look like that at all. The upper block is all scrambled, and there's lots of variation in that. The lower block just looks like a single chromosome had, for some reason, just increased in frequency. And we think that's exactly what did happen, that at this locus, a single allele appeared. It was favorable for one reason or another, and so it increased in frequency, and it happened rapidly. And the reason we know it happened rapidly is because as reproduction happens, chromosomes break apart and are rejoined, and that causes them to get all scrambled up. And this jumbled variation you see up here is the result of a long history of recombination, of crossing over. But this allele down here has increased in number so rapidly that there has been virtually no time for all those recombination events to happen. So we've had this kind of explosion of one allele at the expense of the others. So that is the signature in the DNA that we can look at and that provides us a clue to a history of recent natural selection. Now the next slide is, before you go there, I'll tell you in a second, the next slide we're going to see a kind of large-scale look at this phenomenon. This phenomenon that you see here has a name. It's called linkage disequilibrium, and so you will see that in the next slide. So advance through this to the next real slide. Keep going. Okay. So this now is a view of an entire human chromosome. The upper 3 bars, this bar, and this bar, correspond to different human populations. And the vertical axis on each bar is a measure of linkage disequilibrium in little moving windows along the chromosome. And you can see all these kind of spikes like this. So this spike represents a region on the human chromosome in which there is a lot of linkage disequilibrium, and very likely there was a selective sweep of one allele within the human population of Asia, the top one. This is the Asian population. So this allele increased rapidly in Asia, but not in either of the other two populations. This one over here is the selective sweep that's responsible for the fact that those of us of European descent can drink milk into our old age. So anyway, there are issues here. We'd like to be confident that the number of linkage disequilibrium spikes in this is larger than the number you'd expect to see just by chance, if in fact no selective sweeps had happened at all. We'd like to know, there's a sampling issue to this. Anyway, there are all kinds of statistical issues that I'm going to gloss over and just say that that is an ongoing area of research. The point is, many, if not most of these, probably are recent episodes of natural selection. And next slide. This is a summary from a paper that's now a couple of years old. It's a Venn diagram showing the numbers of selective sweeps that one group estimated in a scan of the human genome. Each circle refers to a different major human population. So these guys recognized 185 selective sweeps in the Asian population, of which 38 were shared with YRI, which is Africa, and 55 were shared with CEU, which is Europe. Only 10 were shared by all three population groups. So the bottom line of all this is that current evidence suggests that, that we are a rapidly evolving species, that we have changed a lot in the last few tens of thousands of years. None of these loci can have been undergoing a sweep that has lasted much longer than a few tens of thousands of years, because if it were older than that, it would have finished by now already and we wouldn't see the evidence. So all of these are recent sweeps. And so we are constantly under ongoing selection at many different sites, and the sites at which we are evolving in one population tend not to be the same as the ones that are rapidly increasing in another. And I'll stop there.
Thank you, Alan. And now we'll move to Don Johansson.
Thanks, Bill. I'd like to start off by thanking Lawrence Krauss and his team for putting this incredible speed learning course together. I've never had so much to learn in one day in all my life. So thank you, Lawrence. Anthropologists are charged with understanding human uniqueness. And as Bill said, in my case, I have focused particularly on the fossil record for human evolution. It assumes, of course, in the title of this session that we are unique. And as biologists, we know that each and every species is unique. But as we think back to Dobzhansky, who wrote, "All species are unique, but humans are the uniquest." Another thing I would probably quibble with a little bit in the title of it is, are we really human yet? When one reads the newspapers, I come away with a somewhat slightly different perspective on what humanness is all about. But by any measure, modern humans and modern human uniqueness has been wildly successful. We are more than 6.5 billion people on the planet today. We occupy virtually every corner of the globe, and we are threatening to occupy and populate other places in our own solar system. As an anthropologist, I think that this incredible success is a consequence of really the unanticipated and powerful synergy of a combination of classical biological evolution with ever-expanding and accelerating cultural evolution. So human evolution is in itself also unique because of the overlay of cultural evolution, which has been so powerful, continues to be, and certainly will be. as highly adaptable organisms, and that is an important clue, as you all know, in biological organisms. It's those that are highly adapted and narrowly adapted that are always teetering on the brink, as Peter Ward told us earlier, on extinction. But as highly adaptable organisms, and I think we are highly adaptable because of this unique combination of cultural and biological processes, we have survived the challenges of environmental and evolutionary changes over time. To understand just how powerful the human adaptation has been, and some of you may have heard Daniel Dennett when he was here, but I was very impressed with the observation and perspective that shows how incredibly powerful this combination of biological and particularly social cultural evolution has been when you consider that 10,000 years ago at the start of the agricultural revolution, I don't know how this was calculated, but the entire mammalian biomass on the planet, which consisted of humans, let me rephrase that. The biomass for humans and their domesticated animals constituted only 0.1% of the entire mammalian biomass on the planet. But within 500 generations of time, meaning today, we now comprise 98% of the entire mammalian biomass on the planet. That has to have important and extraordinary consequences and is, of course, a remarkable reflection of the uniqueness of the human adaptation. Turning very briefly to the fossil record, this has been a great year of celebration for us, many of us, because, of course, it celebrates Darwin's extraordinary accomplishments. His ideas are, as you have heard this afternoon, certainly the core of evolutionary biology, continue to be. I think sometimes my cosmology friends are a little jealous that a retiring Englishman came up with the grand unifying theory of biology without spending a pound of government money on research. Darwin and his great popularizer friend, extraordinary anatomist Thomas Henry Huxley, made a remarkable prediction in their writings in the late 1800s, particularly in The Descent of Man, but also in Man's Place in Nature. And they'd made this prediction without a smoking gun. They did not have a single fossil from Africa. The robustness of the predictability of a theory is in evidence here, of course. They had Neanderthals in Europe. Even Cro-Magnon fossils came a little bit later. But because of the predictive quality of the theory and their keen observation, they predicted that Africa would, in fact, be the crucible where human beings evolved. And today, oddly, in the study of paleoanthropology, where scholarship is accompanied by intense disagreement and controversy, we are now beginning to see, as you will hear from other panel members up here, significant concordance between data from the earth sciences, from archaeology, from paleontology, and much more recently, as you heard in the first presentation, genetics, which is giving us a more comprehensive and, I think, satisfying understanding of the origins of humans and the origins of our own uniqueness. We can now make the following observations about our uniqueness. That were, to some degree or another, part of Darwin's 1871 Descent of Man book. And that was that virtually all of these things I'm going to just touch on were events, occurrences, adaptations, breakthroughs, whatever, that justified the veracity of the idea of Africa being the homeland for humanity. It's generally agreed that older than 7 or 8 million years ago, we shared a common ancestor with African apes, particularly the chimpanzee. We acquired our distinctive form of locomotion, unique among mammals, that is to say bipedal walking, by at least 6 million years ago. The earliest purposeful manufacture of stone tools at about 2.6 million years was an event that transpired in Africa. Our own genus, Homo, arose in Africa, but yet we have only a single upper jaw at about 2.4 million years associated with stone tools for the earliest appearance of our own genus. We know vastly much more about the genus Australopithecus. We know little about how we arose, where we arose, under what conditions we arose, what was the stimulus that prompted that very important fluorescence of a group of different lineages around 3 million years, one of which evolved into ourselves. An important dietary shift from strictly vegetarian to more meat-eating occurred in Africa. Expansion of the brain, for example, somehow associated with certainly social behavior as well as dietary behavior played an influence. But now with the discovery of florist-like creatures, these little hobbits in Southeast Asia, with brains of 400 cc's, what is the relationship between brain size and intelligence? And we know, and Curtis will tell us much more, about the origins of Homo sapiens in Africa that go back about 200,000 years ago. And most importantly, the acquisition of modern human behavior, and I believe symbolic language, arose in Africa. One general overarching question that we should all consider today is the power of place. As was it Janet Brown who wrote the book on Darwin and spoke about the importance of place? The place being Africa, but we still need to understand the nature of that power. I feel that in order to really understand many of the things I brought up, we have to root our explanations deeply in biology and classical natural selection. Humans are not, as cultural anthropologist Alfred Kroeber said, super organic. That view has promoted schisms within the field of anthropology. The view that biology jumps out the window and culture comes in the door is, I think, as dangerous an idea as that of go forth and multiply. Thank you.
Ian?
Thank you. Well, I'd like to start by, again, thanking the organizers for inviting me to participate in this amazing occasion. It has been absolutely wonderful. And I trust that my voice will hold out for the next 7 minutes. It may not matter too much because what I'm going to say overlaps a little bit with what Donna said. But we human beings are very, very distinctive creatures in many ways. And physically, most of our distinctions, of course, have to do with our unique, upright, striding form of locomotion and with the many anatomical accommodations that it's necessitated. But even among our closest known relatives, those whose own ancestors had been bipedal for millions of years, you can see we are highly unusual. On the slide, on the screen right now, you see an image of a Neanderthal skeleton on the left compared to a modern human on the right. And without belaboring the fact, I think that you can see numerous differences between these two creatures, very, very different creatures, particularly in the proportions of the thorax and the pelvis. Anatomically, we sort of came out of nowhere. We have anatomical close relatives of the Neanderthals in the fossil record. We don't really of Homo sapiens. So there's a lot not known right now about where we came from in this context. But it's still true that, as Don has said, all species are at some level distinctive, and it's hardly our physical differences and uniquenesses that give us such a profound sense, I think, that we all have of being somehow apart from nature, of being somehow qualitatively different from any other organism on the planet. And what it is that actually gives us this feeling, I think, is, of course, the peculiar way in which we perceive the world and in which we process information that comes into us from our senses about that world around us. I think alone among organisms, it seems, we human beings are symbolic. That is to say, we're capable of mentally dissecting the environment around us into discrete intellectual symbols. And then we can combine and recombine these symbols in our heads to imagine alternative worlds and to ask questions like, what if? Of course, other animals are capable of responding to symbols. There's no doubt about this. The most famous among these animals is probably the bonobo kanzi, who recognizes several hundred visual and auditory symbols and who can sort of add them to produce and to respond to rudimentary statements. But there's a limit to the practicality of making additive statements. And there's no evidence that Kanzi, the bonobo, is able to manipulate mental symbols in the way that we do and produce an infinity of new meanings by combining a limited vocabulary of symbols. Now, of course, this is not to deny that 90% of what's going on in our heads is, of course, also going on in Kanzi's head. But it really is that last 10%, or whatever the percentage may be, that makes the real difference. Even such prodigiously intelligent organisms as Kanzi live pretty much in the world as nature presents itself to them. While in very stark contrast, we humans live very largely in worlds of our own imaginative making and worlds of our own construction, murky and conflicted, as some of those worlds sometimes are. And it's this intellectual ability, I think, to step back from the world and to mentally disengage even from ourselves that makes our species truly unique. And it's this ability, I think, that most profoundly responds to the question posed today. That is to say, what does it mean to be human? At least to the extent by asking this question, we're asking just what it is about us that makes us extraordinary. What it means to be uniquely human, I think, is essentially to be a symbolic creature. But what, of course, being human subjectively means is to different people is, of course, a different thing. And that is a question to which everybody is likely to have a different answer. And this, of course, is linked to another remarkable aspect of humanity, which is to say that there really is no consistent human condition. You can easily find a human being to illustrate any component of any pair of behavioral antitheses that you might want to come up with. For every saint, you can find a sinner. For every thief, you can find a philanthropist. For every open mind, you can find a closed one, and sometimes all in the same person all at once. And this emphasizes the fact that while our brains are symbolic, they're not entirely rational. And whatever it is that promotes symbolism, it's superimposed on some very ancient brain structures indeed. And this is, of course, because in our evolutionary history, our new way of processing information in the brain has been added to some very ancient functions and structures indeed, rather than superseding them entirely. So the question then arises, how long have our precursors possessed this additional symbolic component, cognitive component that we have? Well, although many commentators, of course, have pointed to evidence for quite sophisticated forms of economic and technological activity far into the past, unlike some of us here, I think, I'm personally unconvinced that most or even any forms of old Stone Age economic activity can be taken as prima facie evidence for symbolism and symbolic activities. And the earliest overt evidence, the next slide, please, of hominid symbolic activity that's bequeathed us by the archaeological record, which is, of course, the record of past behaviors, comes to us only long after the appearance of our species Homo sapiens as an anatomically recognizable entity, perhaps as much as 200,000 years ago. The early Homo that we see illustrated on the screen here seems to go back to about 160,000 years ago. In contrast, next please, more or less unarguable evidence of symbolism is exemplary. Oh, sorry, can you go to the next one in case they're out of order? There we go. In contrast, more or less unarguable evidence of symbolic activities comes in the form of this plaque from a Blombos cave in South Africa, dated to about 80,000 years ago. It's about 100,000 years younger than the earliest evidence we have of modern humans as an anatomical entity. And of course, go back one slide, please. Evidence for the full flowering of human creativity and the symbolic spirit only comes significantly later than this, with the extraordinary outburst of Cro-Magnon creativity in Europe after about 40 or 35,000 years ago. So human symbolic consciousness, which is the underpinning of our cognitive singularity, is thus a very recent acquisition in the human lineage. And even more importantly, it's not a simple extrapolation of the trends that preceded it. Instead, our unique mode of cognition appears to be an emergent acquisition, based on a long accretionary history of brain evolution, but not predicted by that history. And the subsequent history of our species has been marked by the restless exploration and discovery of the evidently many dimensions of that capacity. And I think I've probably spoken too long already. Thanks.
Thank you, Ian, very much. John? Thank you. Pointer. Okay.
Yours
It's
is,
right down there in the
there
bottom.
is John, right?
Right, that's me. Good. Okay, when Bill gave us the task of describing what in your area of research does it mean to be human, I, like many of the other people here, felt this being the 200th anniversary of Darwin's birth and 150th anniversary of the origin of species, I ought to start with Darwin. When Darwin confronted this issue several years later than the origin, he basically just had 2 points to look at. He looked at African apes, which Huxley had shown were the closest relatives of humans, and then he had modern human. And Darwin pointed to a series of features that distinguished humans from our closest relatives, the chimpanzees. As Don had noted, we were bipedal, we walk on 2 legs, we make tools of various sorts, we have big brains, and if you look closely in our mouths, we have fairly small canine teeth, unlike our dogs and cats and other relatives. Next. Fortunately, we're a lot better off these days than Darwin was. As Don mentioned, there was virtually no fossil record of primates or human ancestors in those days. Now we have a lot more intermediate forms, not only between apes and humans, but also of our whole primate fossil record. Next slide. In fact, if you look at the record of all fossil primates, right now there are about 200 genera and over 500 species. I have to say it warmed my heart in earlier sessions when I found out that physicists only recently realized that they couldn't account for 95% of the mass in the universe. Those of us who work in the really hard sciences like paleontology have to live with the fact every day that we're only sampling a trivial portion of life that existed in the past. Nevertheless, working with that material, there are a couple of things we can say about human evolution. Next slide. When people look to see what it means to be human, they come up with lots of features. The stuff that Darwin came up with 150 years ago, bipedalism, our big brains, our use of tools, other, so we say, softer things that don't preserve in bones at least, like Ian talked about symbolism. People look to language. More recently, people look to things like communal child rearing. There are lots and lots of individual features that distinguish us from our African ape relatives. I decided to turn Curtis's question around a little bit and not ask exactly what it means to be human, but how did we become human? Looking at the appearance of, in my case, human anatomy, the morphology of modern humans, following on the quote of Theodosius Dobzhansky, who was mentioned earlier, who said nothing in biology makes sense except in the light of evolution. And I'm going to point out basically 3 features that the fossil record tells us about humans came into being. First of all, the lineage of primates leading to humans goes back over 65 million years. And humans did not suddenly come into existence, but we share features with many, many other of our relatives to varying degree. Next slide. And the transition from either a primitive primate or a chimpanzee to a human has clearly not been a simple linear progression. We often see it shown this way, in which there's a gradual change from this quadrupedal animal slowly getting a little taller and a little taller and a little taller, and its brain's getting a little bigger and a little bit bigger, and its teeth are getting a little smaller and a little bit smaller. Rather, that's not the case, because humans today, in fact, are a whole mosaic. Some of the features in our anatomy, such as, for example, our fingernails, go back to the origin of primates. We, like all primates, have fingernails. Our As Don mentioned, our pelvis goes back over 3.5 million years. Our bipedal behavior is certainly 3, 4, 5, even 6 million years old. Other features like our big brain came much later. We have a dental formula, however, with all living old world monkeys and apes that goes back 35 million years. We're a complete mosaic of different features. We didn't just appear full-blown, and things didn't appear slowly all together. Some came early, some came late. One consequence of this is that there are a lot of contingencies. The appearance of human things that are important in human behavior and human evolution didn't appear in a random sequence. As Don Johansson mentioned, bipedalism goes very, very back in our lineage to 4, 5, or 6 million years. It's not a coincidence that the appearance of stone tools was after our ancestors were already bipedal and had their forelimbs already freed. At the same time, those hands that could make and use stone tools were using an elbow that we share with apes that let us move our arms in lots of different ways, was a hand that had an opposable thumb, something that we shared with all old world monkeys and apes, and also was a hand that had fingernails, which are primate features. It would have probably been virtually impossible to evolve tool use like this with all these antecedent features hadn't already been in place before we got to that stage. So human evolution took place, the appearance of modern humans took place step by step, but not either a continuous smear or a random series of events. And finally, whether we look at primate evolution or human evolution or probably the evolution of virtually all organisms, in retrospect, human evolution obviously wasn't the kind of continual progression we often see it portrayed, but it involved lots and lots of what, in retrospect, look like false starts. We're very successful animals in the Eocene, for example, but are only represented by very, very few relatives today. There were lots of major radiations that have been snuffed out, leaving only bits today. Even in human evolution, next slide, we know that our ancestry, again, as Don mentioned, consists of huge radiations, a whole radiation of animals down here who were all bipedal. But of all these bipeds, not all of them went on to develop stone tools and big brains. And of all those that seem to have used stone tools and big brains, only one of them survived to become us, the symbolic creatures. So there's lots and lots in human evolution that we know about that Darwin didn't know about then. Next and final slide. So I think the question we're all trying to get at is not just what it means to be human, but now that we have a time depth, to learn exactly how we came to be the way we are. Thank you. Thank you very much, John. And we'll finish up now with Curtis.
So I can find. Let's see if I can find myself here. There we are. Okay. I'm going to bring us up to the final leg of human evolution, at least to the origins of the modern human lineage. Genetics, fossils, and archaeology and climate must be integrated to tell the story of the origins of our species. And at the bottom of the slide, I've provided a proxy measure of the global climate record change over the last 750,000 years derived from the recent Epoca ice core from Antarctica. John, could you pass me the pointer there?
Oh, sorry.
Okay. And it's pretty self-explanatory. The key pattern here is that when the line is low and it's in the blue, we're in a cold glacial, and when it's high, we are in a warm interglacial. And note that the amplitude and thus the abruptness of climate change increases over time, and particularly after 400,000 years ago, we see dramatic amplitude. And for the most part, at the latter part of modern human origins, we were in a glacial phase, so climates that are quite different than what we see today. Now, the recent genetic evidence suggests that gene flow between the two major lineages that we have in the fossil record, Neanderthals and modern humans, was cut at about 400,000 years ago, and they began two distinct evolutionary paths. Homo sapiens evolved in Africa, and the first fossils that we find that are assigned to Homo sapiens are from Ethiopia and North Africa, and they're dated between 200 and 150,000 years ago. And it's important to recognize that these particular fossils are classified as Homo sapiens, but may not be directly on the genetic lineage that leads to modern humans. The most recent genetic data puts the origin point of the human lineage as occurring between 190 and 140,000 years ago. So the lineage that leads to all modern humans that are alive today appears shortly after the earliest Homo sapiens fossils do. So the fossil record and the genetic record are in sync. This graph once again shows a proxy measure of global climate change, and I draw your attention to the period between 200 and 123,000 years ago. The period just before the origins of Homo sapiens is an interglacial that we call marine isotope stage 7, or MIS 7. It's followed by MIS 6, which is one of the longest, coldest glacial periods on record. Note that the estimates for the species origin point that I mentioned earlier fall at the beginning of MIS 6. For some time, it has been known that modern humans have extremely low genetic diversity, and several studies attribute that to a tight genetic bottleneck. The Fagundes study, cited at the bottom here, puts that bottleneck down to an effective population of 600 people at approximately 140,000 years ago. This progenitor population existed during the long, cold MIS 6 glacial that I just talked about. The African continent is cool and dry, poorly vegetated with widespread deserts. There would have been many natural boundaries to movement and gene flow at this time. Gene flow gets cut and the lineages diverge, and I suggest that we have 4 to 6 potential progenitor locations. I think the origin population is probably on the coast of South Africa, and that's not only because that's where my field work is. The splitting points of the ancient African lineages relate the story of the importance of these glacial stages that we've just talked about. If we take the divergence times of these lineages, which I have plotted here, and these are the ancient African lineages composed primarily of extant hunter-gatherer groups that we have today. If we plot the divergence times, which I have done here, you will see that divergence times tend to fall on glacial stages. Glacials drive lineage formation near our origin point. The last 20 years of research in Africa documents a jump in the evidence for innovation and symbolic behavior at about 70,000 years ago in Africa, and Ian was briefly mentioned some of the African archaeological evidence for that. We don't see a jump in that innovations and symbolic activity in Europe until about 40,000 years ago, and that is almost certainly due to the migration in of modern humans who bring that cognitive capacity with them. Our recent work on the south coast of South Africa suggests that the origin point of these innovations in symbolic behavior likely stretch far more back in time than that 70,000 point. And I think the reason that we have been missing these sites is because we have very, very few archaeological sites in Africa that date to this time. At this point, we have about 3 sites in Africa that have been dated to MIS 6, almost certainly because populations were very low and dispersed, and it's hard to intercept them in the archaeological record when that is the case. Modern humans originated during a grim glacial climate crisis that isolated us into small populations. Glacial pulses drove further early diversification. That genetic bottleneck is a final, nearly catastrophic black swan event that sculpts our origins. It wipes out much of the genetic diversity that our species had accumulated over a very long time. It sets environmentally driven phenotypic diversity to nearly zero. It establishes the progenitor population, its local peculiarities, and sets a baseline for all modern humans. The one population that survived made a cognitive leap to the ability to create novel associations between seemingly unrelated phenomena. I think the most parsimonious explanation is that jump occurred at the point of the origin of our lineage, around 160, 170,000 years ago. Those novel associations are the so-called genius moments that result in strong fitness benefits when they are translated into technologies. They also allow the evolution of symbolic behavior. That allowed innovations to be taught, passed on, and improved. I think that population was on the coast of South Africa. The reason is that the rich shellfish beds that we find there and the super diverse tuberous plant foods provided a rich diet of protein and carbohydrate during this grim climate crisis. Coastal diet allowed increased group size, decreased mobility, and put a premium on cooperative behavior. Our origins are tied to the sea, and the baseline adaptation is life on the beach.
Thank you, Curtis. We have about 10 minutes for questions, and I will ask one question of the panelists, and then we'll turn it over to general discussion or questions from the audience. And my question to each of you, and I would like a fairly concise answer if you can, is, from your perspective, what is the single major gap in our knowledge that impedes a richer, more complete rendition of human evolution? And let's just start down at the end with John and work our way up.
Well, I mean, I could give you the fake answer. The single gap is most of the fossil record.
Well, don't give us the fake answer.
I think the biggest gap that we all would like to see is the gap that we still have, even though we have a fossil record going back to 5 or 6 million years. There's still a long distance between that and our closest living relative, chimpanzees. And, you know, we still don't know what's going on back there, and I think that's the one that would make things a lot easier.
So a fossil record of chimpanzee evolution, for example.
Yeah, and closer to the chimp-human divergence.
Yeah. Alan?
From the genetic perspective, I can think of two that I'm afraid I have to say both of.
Okay, speak quickly.
One I've already alluded to, and that is the fact that what we really need is a set of polymorphisms that were sort of chosen at random rather than chosen in a sort of mysterious way. And we should have that by the end of the year with the 1,000 genome projects that's ongoing. The other thing that we need is a better way to calibrate the mutation rates so that we can get accurate dates out of the genetic differences that we can actually measure. And what struck me in listening to this today was that we can turn Curtis's work on its head and use it as a calibration of the mutation rate rather than as a kind of an interesting result. We can use it as the starting point of our analysis, that most of the coalescent events ought to happen during those periods when the population size was small, and that those ought to be during periods when the climate was cold. And so that may give us a calibration that's much more useful than the traditional chimpanzee calibration.
Curtis?
I'll answer the question in regards to the material I just spoke about, specifically the origins of modern humans. The biggest gap that we have right now is simply a lack of field work. If you take that map that I put up there that shows where those hypothetical progenitor populations are, in order to test the hypothesis that I laid out, we have to have intensive field work in those 4 to 6 localities. And right now I'd say that there's reasonably good field work going on in 3. And the basic problem is logistics, money, people who are willing to go out and do that kind of field work, which is extremely difficult. So if we could put 50 years of time and money into it, I think we could actually come close to answering those particular questions.
Great. Don?
I think the most intriguing gap is the gap between about 2.6 million and 3 million years ago, when we saw the stage set for the origins of a genus, our own genus, genus Homo, that would have widespread effect on the entire world. We know very little about it. We have inklings that climate played a very important role, but the link between Australopithecus and Homo is one of the most important missing gaps, I think, in the entire human fossil record.
Thank you. Ian?
Thanks. Well, John started at the very early end of the time scale, and Don has gone back to that. I'd like to go, like Curtis, to the very late part of the time scale. I agree completely with Curtis that symbolic behavior was born, or the potential for symbolic behavior was born with the with the genetic reorganization that gave rise to Homo sapiens as a anatomically recognizable entity. What I'd like to see developed is some way of being able to read from the archaeological record exactly when symbolic behavior started. Symbolic behavior and complex behavior, not necessarily the same thing. And I think we're in basic agreement, but I don't think we'll solve this until we really have a good measure of symbolism in the archaeological record.
Great, thank you. And now from the audience, question over here.
A question for Curtis. I'd love to have you elaborate on what you say there. Our origins are tied to the sea. In particular, for the long list of features that make up the aquatic ape hypothesis, crying, fat distribution, dive reflex, could you be specific? Which features of human anatomy and behavior do you think were actually shaped by our ability to get food from the sea?
I wouldn't say so much that our morphology and anatomy and so on is shaped by an aquatic adaptation. The aquatic hypothesis actually goes way back further in time than what I'm arguing. What I'm arguing is that during marine isotope stage 6, during this climate crisis, the ecological conditions in the Cape, which I could go on for an hour about, were perfect as a refugia area for modern human origins. And when people broaden their diet to marine resources, and we've documented the earliest evidence for that, it's at 160,000 years ago at Pinnacle Point in South Africa. What we see time and time again is they reduce their mobility, they increase their group size, and that puts added strains on mechanisms for mediating social relations, which of course is one of the hallmarks of modern humans. And so there's kind of a potentiality argument to part of it. The other part is that shellfish and marine resources are very high quality diet with very high omega-3 fatty acids, which are critical for brain development, placental development, and so on and so forth. And I'm not saying that, some people have argued that the terrestrial food chain does not have enough omega-3, and I wouldn't argue that at all. But what I would argue is that once you broaden your diet into the marine food chain, you release the need to work hard to get omega-3s from the terrestrial food chain. And that's a benefit. And also it has fitness benefits because the diet is very high quality. So I don't think there's anything, I don't think there's kind of a surfing morphology or anything, but I think that sea adaptation is one that's perfect incubation for the modern human uniqueness that we see.
Another question right here in the front? Oh, sorry, go ahead. Sorry.
Yeah, so this has two parts. The first is, I don't know if this is working, but it concerns me that there's such a time gap between the flowering of art and symbolic expression, as you showed us, and the origin of anatomically modern humans. And I'd like to know what the panel reckons is the reason for that. And two, in these glacial times, of course, we're looking at sea low stands. And is it on the to-do list to do some underwater archaeology?
Do you want me to? Well, I'll just to firstly address the second one. Off South Africa is the Agulhas Bank, which is a very slowly descending continental shelf. And we've used bathymetric data to develop a 3D model of that. So the coastline, for example, at 150,000 years ago was 90 kilometers out. And I think that population was out at the intersection of the coastline. You can't do underwater archaeology in that area. It's just logistically too challenging.
Did you have a...
I would just say that it probably has to do with the lack of discovery, in the sense that stone tools come in at about 2.6 million. Well, obviously they were being made before, but how much before we don't know. So if you conclude that they must have appeared a couple of 100,000 years earlier, there's not that much a gap between Australopithecus and early stone tool behavior. Australopithecus may have in fact made tools. And in the state, with regard to later sapiens, we have things from Curtis's site, which he didn't really mention very much, were the ochre pencils that are used in high numbers in strata that are as old as 160,000 years. Even if they painted on those cave walls would not have preserved the art that you have in Europe, such as Ian showed, which are guarded because they're deeply underground. And it may be that the origins of modern symbolic behavior, as evidenced in this marine isotope stage 6, back as much as 160,000 years, would have only separated us by about 30 or 40,000 years from the appearance of Homo sapiens. So we may really reduce the difference between, or the time span between the origins of Homo and the origins of symbolic behavior.
One more question right here in the front.
Just to remind a possible interesting gene section to look at, and that is about 8% of our genome is made up of viral homologies, more than the number of reading frames. And these genes have entered, that is the viruses, have entered the genome at different periods, and probably in very ancient times. It's interesting to consider if a wide epidemic might have had some impact on the human changes.
I don't have a response. As you say, it's interesting, and I don't have any comment.
Yeah, thank you very much. I do have three things to say. First of all, so we've ended at 5:05, and that means the bus is for those who are going to dinner, and you know who you are. It's at 5:35, so I want to give you half an hour. The second thing is that there are actually posters of the galaxy, the Hubble Deep Field, very expensive posters that were made that you're welcome to take, thanks to the courtesy of Roger Windhorst. And the last thing is, while my colleague Don Johansson said it, I was going to say it tomorrow, but I might as well say it now. You should not suffer from the delusion that I organized this meeting. You will see a list of people here, and these are, yeah, I know, but each of whom, they're not courtesy